Hello, hello. Today we're going to be doing the lectures for Lab 9 and Lab 10 in one sitting because they're both relatively short. Please forgive if my voice cracks or anything. All the changes in weather lately have made my allergies just a whole pain. But let's get started. So first of all, we're going to be talking about anatomy and diversity, specifically in primates. Some key terms, we've talked a little bit about these before, but just to kind of reiterate, strepsirini, these are going to be lemurs and lorises and other monkeys that have moist noses. Haplorines are going to be tarsiers and simians. They're going to have dry noses. Within haplorines, we have platyrrhines. These are going to have flat noses with sideways facing nostrils. These will be tamarins, spider monkeys, capuchin monkeys, cebidae, and squirrel monkeys. And then catarrhines, these are going to have hooked noses with nostrils facing downwards. So your old world monkeys, the great apes, baboons, the greynon, macaques, and even humans. Then cercopithecoids, this is a family of mammals, that means old world monkeys. Hominoids, which is a primate group that includes humans, our fossil ancestors, and the anthropoid apes. And then hominin, which is part of the family or the larger group of primates called hominids. Now, hominids include orangutans, gorillas, chimps, and human beings. However, hominins really only include specific human ancestors and then humans. It's kind of the square and rectangle thing that you learn in math. All hominins are hominids, but very few hominids are hominins. Here you can see a little bit more about the differences between strepsirini versus haplorini. And then this little cladogram here kind of shows you how everything branched off. Now we're going to go over some key terms about dentition. Most of these aren't super important for you guys to know, but they are leading up to something, I promise. So to start off, we have enamel. This is that kind of hard, glossy substance that covers the crown of the tooth. This is what we try to protect by brushing our teeth. Then we have cementum, and this is a very thin layer of bony material that helps fix your teeth into your jaw. Now, a lot of times when forensic anthropologists get human remains, they are oftentimes missing teeth. And this is because whenever you die, your cementum actually erodes away. It does this even quicker if remains have been found outside where they have been exposed to different weathering conditions. Without that cementum, your teeth get very loose. It's very easy for them to fall out over time. Then we have dentine. This is a very hard, dense, bony tissue that forms the bulk of your tooth and sits beneath the enamel. Crowns are the part of the tooth that actually projects from the gum, so it's the part that you are actually brushing that you can see. The neck is a narrowed part of the tooth between the crown and the root, so it constricts just a little bit. The root is the lower 2/3 of a tooth, and they're normally buried in bone, so either in your maxillary bone or your mandibular bone, and they help to kind of anchor your teeth into their position. The occlusal surface is the portion of your tooth that comes into contact with another tooth in the other jaw. So it's basically just that top of your teeth that you use whenever you bite or chew something. Cusp. These are these little bumps that you see in teeth. It's an occlusal or incisal eminence on a tooth. Canine teeth, sometimes known as cuspids, each possess a single cusp, whereas premolars are sometimes known as bicuspids because they typically possess two cusps. Now, molars often possess either 4 or 5 cusps. Sometimes you will see molars that have more cusp. This is sometimes due to medical issues, as well as just other genetic factors. Occasionally, you will see molars that have less than 4 cusps, but that's a little bit more rare. But the average human, quote unquote, has 4 or 5 cusps. And then the crest is the maxillary ridge that passes along the alveolar process of the fetal maxillary bones. Basically, it's that kind of bumpy part on the top or roof of your mouth. If you feel it with your tongue, you'll feel where there's kind of like a line. And that actually fuses as you get older. Now, what we're building up to is talking about dentition formula. So you can actually count the dental formula for any animal. Humans have a 2, 1, 2, 3 dental formula. And the way that you count these is that the first number is counting the number of incisors. The second is your canines. Third is your premolars. And then finally, the fourth is your molars. Now you might want to argue that humans actually have 4 incisors and 2 canines and 4 premolars and 6 molars. But that would be a lot. So we actually just count half of the teeth. So you literally split it in half and count just one side of the jaw or the maxillary surface. Like I said, humans have a 2, 1, 2, 3 dental formula. That third molar is actually your wisdom tooth. Now, it used to be that everybody had wisdom teeth pretty much, and it was because we were eating much harder foods that wore our teeth down very quickly. And so you would get a wisdom tooth later on in your life than you would the rest of your adult teeth. That would kind of help replace the molars that had worn down so that you could still continue to eat. We also had wider jaws back then. However, as humans have started to eat a much more processed and soft food diet, our jaws have gotten smaller. Because of this, we don't have as much room for third molars, but we also don't wear down our other molars as quickly or as severely as our ancestors did. So that third molar isn't really necessary anymore. It's actually a very interesting case of evolution still occurring as well, because humans are actually evolving to have wisdom teeth less often than we used to. So every year we're seeing people born that never develop wisdom teeth at all. Now, the fact that our jaws have gotten smaller have also meant that we don't have as much room for wisdom teeth, which is why if you do have them, people typically get them removed because they crowd and we just don't have the space for them, so they cause other issues in our mouths. Now, not all primates have this 2, 1, 2, 3 dental formula, and not all primates have a dental formula that matches between the top and the bottom teeth. You will see this on the homework for this week as well. But we will go over it very quickly together. So when you're trying to decide between if something is a non-primate or a primate, you can tell if they are or not, because all primates have a bony rim that completely encircles their orbits. This is the eye orbital. All primates also have forward-facing orbits. So we have forward-facing eyes. And then we have a relatively large brain compared to non-primates. Now, as we get more specific, the differences between strepsirrhenes and haplorrhenes. Strepsirrhenes tend to have longer snouts and a greater reliance on smell. They often have tooth comb incisors, These are just very long, thin, closely set together incisors that are actually used to kind of comb through fur, comb through leaves to catch insects. So it's also a good way of telling if something is an insectivore or not. If it has a tooth comb, it is likely an insectivore. Strepsirrhines also have an ectotympanic ring, while haplorhines have an endotympanic ring. Strepsirrhines are often nocturnal, whereas haplorhines are usually diurnal, so they are awake partially during the day and partially at night. Haplorhines also have a fused mandible, as well as an enclosed eye socket. So this is that enclosed eye orbital. This is why in humans we actually have where our eye sockets sit, we don't have an opening at the back. It completely encloses our eyes. All haplorhines have this. A lot of strepsirrhines don't. This is also sometimes known as a postorbital plate. Now, between tarsiers, platyrrhines, and catarrhines, catarrhines have downward pointing noses, whereas platyrrhines have outward pointing noses. Platyrrhines also have prehensile tails, whereas catarrhines, if they do have a tail, it isn't prehensile, it tends to be shorter as well. Or more often, they just don't have a traditional tail at all. Catarrhines have 2 premolars and that 2-1-2-3 dental formula, just like humans. Platyrrhines have 3 premolars and a 2-1-3-3 dental formula. Now, tarsiers, I will mention again, they get a little bit weird. So they actually have a 2-1-3-3 dental formula on the top, or their maxillary dental formula, and then a 1-1-3-3 mandibular dental formula, or bottom. They also have very large eyes relative to their brain size. Incredibly large eyes is indicative that something is nocturnal, as well as that it is an insectivore. Now, between Cercopithecus and hominoids, hominoids tend to have relatively larger brains than Cercopithecus, as well as having 5 cusps on their lower molars and a Y5 fissure pattern. Now, you can look this up, but essentially it just means that in the middle of a molar, there is kind of a Y shape that you can see, and it's what helps divide that molar up into 5 cusps. By the way, humans typically have a Y5 fissure pattern, but there are actually a couple different fissure patterns that humans typically have. Y5 is just one of the most common. Now, between hominoids and hominins, hominoids tend to have lower third premolars that are single cusped. They also have lower faces that project more than hominins, so they have more facial prognathism. They also tend to have larger canines that are honing, so it is more of an indicator of sexual dimorphism that hominins don't typically show as much. Hominoids also have relatively smaller brains compared to hominins. Once we get to hominins or early human ancestor, this is when we start to see brain capacities really shoot up, which makes sense because this is also when we start to see more refined tool making, more refined fire use and cooking of food, as well as structure making and social groupings. And then hominoids have a foramen magnum that's placed much further underneath the brain case. The foramen magnum is actually this little hole at the bottom of your skull that is where your spinal cord kind of helps attach your head to your body. Now, as something becomes more bipedal, we actually see that foramen magnum move to be more centrally located underneath the skull rather than further back, which we'll talk about in the next lecture as well. Now, when you do the homework, or Lab 9, I should say. You guys will need to go to this eSkeletons website. It is also linked on the additional resources folder on Blackboard. You have a little bit of freedom on which primates that you choose to look at through this. But essentially, you will be choosing a couple of primates. You will be looking at their skulls and their dentition, and then trying to determine if they are primates or not, whether they belong in the Strepsirini or Haplorhini group, then whether they are in the Platyrrhines or Catarrhines group or tarsiers, the Cercopithecoids or the hominoids, and then if they are hominins or not, and why. So you'll have to give a couple reasons, a couple traits that helped you place them into each category. Now moving on, we will talk about Lab 10. Which is about the evolution of locomotion, specifically about bipedalism. So some key terms, we've talked about this before. Locomotion just means the movement or the ability to move from one place to another. Bipedalism is using only two legs for walking. However, there are different types of bipedalism. So there are obligate bipedalist organisms. These are going to be the form of bipedalism that's assumed to be regular or habitual. So what a mammal is consistently using as their form of locomotion. Very few mammals today actually demonstrate habitual bipedalism. Some good examples of it, though, are humans and kangaroos. The other type of bipedalism is facultative. This is an animal that is capable of walking or running on two legs, but it's typically a response to kind of exceptional circumstances, and they don't do it as their primary form of locomotion, so they were normally walking or running on four limbs or more. For example, lizards and other primates. We talked last week about chimps and bonobos. Of the great apes, they tend to walk bipedally more often than the others. However, gorillas and orangutans and even baboons sometimes do walk bipedally. Chimps and bonobos do it a little bit more often, with bonobos doing it the most often of primates outside of humans, but it's still not often enough for them to be considered obligate bipedal creatures. Now, we've talked plenty about the planet of the apes during the Miocene. However, by the late Miocene, a good majority of these ape species go extinct. This is partially due to competitive exclusion, which states that if all other ecological factors are constant, then two species competing for the same resources cannot coexist. Now, this doesn't mean that two species in an ecosystem that use the same resources occasionally can't coexist, obviously, or else other, entire ecosystems would fall apart. This means that two species that are completely reliant on the exact same resources cannot coexist because there just are not enough resources to sustain that. Many primate species died out because of this. However, a lot of them also just could not adapt as the environment was changing radically during this time as well. However, the late Miocene is when we see the first evidence of bipedalism in the family Hominidae. We see this in Australopithecus. Of course, this is sometimes debated because there are earlier human ancestors that show some signs of bipedalism in their skeleton. However, it isn't enough to say that they were consistently or habitually walking bipedally. Australopithecus is the first species that we can say definitively was walking bipedally a good majority of the time. Now, here in the photo on the left, you can see the remains of an Australopithecus individual. She's actually nicknamed Lucy, and she is one of the most complete skeletons that we have of Australopithecus. As such, she is also the typecast for the species. Here in the middle, you can see where they have kind of 3D printed and emulated what her skeleton would look like if we had all of it. It's fairly human-like. Some differences do include that her arm and leg ratios are longer than modern humans relative to the trunk of her body. Her rib cage is also quite a bit wider than modern humans. And then her pelvis, or her os coxa, we can definitively say that she was bipedal because of the way that her femur kind of sits into her os coxa, as well as the shape of her os coxa. It's much shorter and rounder or wider, like modern humans, than it is tall and long, like a lot of other primate species. Her femur also angles slightly inward, which is a characteristic of bipedalism, and it's one of the things that helps us stand upright and be able to balance correctly. You guys should watch this video about Lucy. We can also tell that Australopithecus was bipedal due to the finding of the Laetoli footprints. That is what this photo on the right-hand side is. Here you can see there are actually 2 sets of footprints. There is some from an adult, and then some little tiny ones next to it that likely belong to a younger child. Of course, there are a lot of emotional stories that people have kind of conjured to explain these. But you guys should watch the video linked here about the Laetoli footprints as well. You can really tell here that the foot was designed very similarly to how our feet are designed. And by the way, human feet are very unique as a result of our bipedalism, in the way that they are structured so that we can balance and the way that we distribute weight. But you can see here that these are very similar to the way that our feet or our footprints would be shaped, as well as the distance between each step is very similar to a modern human gait. Now, I've talked about the femur kind of setting into the os coxa and how that indicates bipedalism. This is because of the morphology of the acetabulum, which is that socket that your hip actually, or really what most people would consider their hip, even though it's not. It's where your femur kind of connects and articulates to your os coxa. We can also see some changes in the sciatic notch and the sacroiliac joint, as well as the pubic symphysis and the iliac blade as primates become more bipedal. The pubic symphysis becomes kind of wider, the iliac blade becomes wider and shorter rather than being tall. It's also not quite as sharp as it is in earlier primates. We also see changes in cranial morphology as our ancestors became more bipedal. So here on the left-hand side, you can actually see the Australopithecus skull. On the right-hand side, we have the Homo sapien. There are some pretty obvious differences. In between, we have Homo erectus, which is the first Homo species, as well as Homo neanderthalensis. Now, before we were as bipedal as we are currently today, we see more sagittal crest. This is a ridge of bone that runs along the midline or the top of the skull. Humans have a sagittal suture. We don't have a sagittal crest. However, our early ancestors, especially Australopithecines, Paranthropines, even Homo erectus, they do have sagittal crest, and it's fairly obvious when you see it because it is kind of just like a bony ridge up there. It's pretty exaggerated in some species. We also, as we become more bipedal, we lose some postorbital constriction. And this is the narrowing of the cranium just behind the eye orbitals. It's found in most non-human primates, as well as early hominins. Homo sapiens technically do have a little bit of constriction there, but it isn't extreme enough to really call it a postorbital constriction. You'll see what I mean. I'll show you some other photos at the end of this where you can see what postorbital constriction really looks like, because it's very exaggerated. And the reason that we lose this as we become more bipedal is actually because as we become more bipedal, we see increased brain sizes. And as we move to more modern humans, so when our more modern ancestors, so Neanderthals and modern humans or Homo sapiens, we also see increases in brain size, specifically in that prefrontal cortex, so that kind of forehead area, which reduces the postorbital constriction. We also see more facial prognathism in earlier ancestors that were less bipedal. And this is just the extension or kind of that bulging out of the lower jaw, so the mandible. You can see in this left-hand photo of the Australopithecine that the jaw angles out from the eye orbitals quite a bit, whereas when you look at Homo sapiens, it is fairly straight. So again, when we are trying to recognize bipedalism in the fossil record, we're going to be looking at the skull or the cranium. Along with the things we just mentioned, we're also going to look at the placement of the foramen magnum. This is where the spinal cord connects. You can see here that in dogs, it's fairly far back because dogs are walking entirely on four legs. In chimps, we see it move a little closer to the center. This is because they walk at more of an angle with their knuckle walking. But once we get to humans, we see the foramen magnum very centrally located underneath the skull. This is because to stand upright, your spinal cord has to connect a much straighter line. And it also helps us to be able to move our heads in different directions, which is helpful because we do have forward-facing eyes. So it gives us a wider range of eyesight, as well as stabilizing our head while we walk upright, because you need a lot more balance to be able to do that. We also see changes in the spinal column, so the curvature of the spine. Here you can see the chimp has a fairly straight spine. I mean, it's angled, but it's straight. Humans have sort of an S-shaped spine, and this is actually to kind of help lower the impact that our spine takes from walking upright, as well as helping with like our center of gravity and things like that. We also look at the pelvis. You can see the chimps are fairly long and thin. Australopithecus is kind of somewhere in between, but it does resemble a human more than it does a chimp. It is wider, it is more bowl-shaped. And then humans have wide, short, and very bowl-shaped pelvis. So we have a much larger area in between each os coxa, and it angles forward more instead of being very straight. We also look at our knees, so they angle in from the hips more as we are bipedal. That helps to reduce the weight on our knees, but also with balance. And then our feet also change. We have big toes that are bigger, they're also sturdier, and they're more in line with our feet and very uniquely designed, as I mentioned before. We also tend to have more of an arch in our feet. Now, obviously, there are some benefits to being bipedal. It makes it easier to pick up food and other resources. It frees up our hands for carrying things, including babies, and improving our fine motor skills, which allowed us to create more tools and use them. It also enabled early humans to appear taller and larger and more intimidating, so that they weren't just being picked off by better predators. It also helped us cover wide open landscapes more efficiently and quickly. Alongside that, it allowed us to be built more for endurance, resulting in long distance hunting strategies. So one of the earliest ways that humans actually hunted was basically by tiring other animals out. Obviously, we're not as quick as a lot of other animals in terms of just straight speed. However, humans are endurance animals. We are actually very optimally built for endurance, and part of our brain size is to do with that as well. But early humans would essentially chase other animals. Eventually, you stop, actively chasing them, as in running at them, and you just keep following them until that animal collapses in exhaustion, and it's pretty easy to pick off. Humans, it's not that we don't get tired, but we can force ourselves past that. Now, with all the benefits that come with bipedalism, there are also costs that we still feel today. So part of being bipedal means having back pain. You feel a lot more shock up in your spine. Standing upright all the time actually eventually compresses the lower disc in your spine. It's also why a lot of older people tend to get arthritis in their backs, as well as their hips and their knees, because it puts a lot of pressure on those areas as well. And a lot of times in older skeletons, when we're looking at them, not only do you see arthritis, but you can actually see in a lot of the areas of the skeleton that are more mobile, so your hips and your knees, your shoulders, a lot of those areas actually end up rubbing down and rubbing away bone eventually, because you lose a lot of the cartilage in them as you get older. We also see more instances of scoliosis, as well as slipped disc in the back, and collapsed foot arches and other related foot problems. Now, this is because evolution doesn't actually care about a species. It's not a sentient thing. And you can't really control evolution in your own species. You can kind of control it as far as like propagating and, breeding for very specific genetics, but long-term evolution, you can't really control like that. And evolution kind of just makes you good enough to survive. It doesn't actually make you the most optimal that you can be as an organism, because it doesn't care. All it really cares about is you continuing your genetic line, and you don't have to be perfect to do that. Now, humans are still evolving, so maybe in the future we will see less instances of some of this stuff, but it will be a long time before that is the case. You should watch this video on the evolution of bipedalism. ignore these due dates. And I am going to show you just a couple of images to kind of go over. So here you can see that sciatic nosh that we talked about. The ilium and the iliac crest, that's this kind of area over here. And then the acetabulum, this is where your femoral head fits in. Like I said, as people get older, this like ridge here tends to wear away because walking bipedally actually does break down a lot of your body as you get older. Moving on, this is that sagittal crest I was talking about. It's fairly obvious. It kind of looks like a fin to me on the top of the head. Humans, modern humans, do not have sagittal crest. We have a sagittal suture, and sometimes you can see that somebody has sagittal keeling, which is where their sagittal suture is raised a little bit, or kind of angled more like an upside down V. But it is not a sagittal crest. It is never that extreme. This is that postorbital constriction. So while modern humans have a slight kind of divot behind their zygomatic arches, which are these areas right here, it is not actually postorbital constriction like we see in other early human ancestors like Paranthropines and Australopithecines. This is very obvious. You can see there's not a lot of space in between here. There's not a very large prefrontal cortex in the brain. And there's a lot more space between the zygomatic arch and this kind of edge of the eye orbitals. Now this is that prognathism. You can see it a little bit more. You can see here with extreme prognathism, you see a very sharp angle. As we get more bipedal, this reduces until we get to modern humans, which is relatively straight. There is a slight kind of change. It's not perfectly straight. And some of this is actually dependent on ancestry. So some ancestral groups have slightly more prognathism than others, but it's not anywhere near as extreme as what we see in our other human ancestors, especially in our early human ancestors. And then this is that Y5 fissure pattern in the teeth that we talked about. Here's this kind of Y shape, and then 1, 2, 3, 4, 5 cusp. There are some other ones, as I mentioned, but Y5 is one of the most common to see.